Hello, everyone, and welcome. Thank you so much for joining us today. I'm Faye Hobbs, the Research and Patient Involvement Manager at Ovarian Cancer Action, and we're the leading ovarian cancer research charity in the UK. And because research is such a major focus for us, it enables us to support ambitious long-term projects like OvarianVax. I also lead the patient and public involvement work for the programme. We're really excited to bring this session to you today because this is the first time we've opened up a wider public conversation about OvarianVax and what it could take for something like this to become a reality. So the session today is about two things. Firstly, sharing some of the early progress from Ahmed's work so far, and then opening up a wider conversation about what it would take for a preventative cancer vaccine to succeed in the real world. And I'm really excited to have Professor Peter Sasieni join us today. I followed his work around HPV vaccination and cancer prevention for about 15 years now. So I'm genuinely really excited to have him as part of the discussion. And also because his perspective goes beyond the science itself and into what it actually takes for prevention to work at population level over time. So before we begin, a quick note on where the project currently is. OvarianVax is still in the preclinical stage. That means the vaccine's not yet been tested in people, and there are still some important scientific and regulatory steps ahead. But what makes this project so exciting is that it builds on more than a decade of Ahmed's research into how ovarian cancer begins, and particularly discoveries showing that many ovarian cancers start in the fallopian tubes and that the immune system may not be able to recognise some of the very earliest abnormal changes. And what the team is now trying to explore is whether the immune system could potentially be trained to recognise and eliminate those early changes before cancer develops. So before Ahmed kicks off, I'll introduce both of our speakers today. Ahmed is Professor of Gynecological Oncology at the University of Oxford and leads the Ovarian Cancer Cell Lab. And his work over the last decade, like I just said, has helped to reshape how we understand the early stages of ovarian cancer development and laid much of the scientific foundation for OvarianVax. And I'm also delighted to introduce Professor Peter Sasieni. He's a Professor of Cancer Epidemiology at Queen Mary University of London and one of the UK's leading experts in cancer prevention and screening. He's played a major role in research around HPV vaccination and the cervical cancer prevention programme, and today he's going to help us think more broadly about what history can teach us about prevention. So he'll include what helped programmes like the HPV vaccination program succeed, but also why some prevention approaches struggle to translate into widespread uptake and long-term impact. So over to you, Ahmed. He's going to give us an update on where the program of work is now.
Thank you very much, Faye, for the introduction. So I'll just start by explaining our efforts in producing a vaccine for prevention of ovarian cancer, and just really start from the basics of explaining how a vaccine in broad terms has two components in its development. One is the actual payload of the vaccine, that is comprised of different, what we call epitopes, that represent proteins of the entity that we are trying to prevent against. So if think about COVID vaccine, for example, you would have proteins or portions of the protein of the COVID virus that we try to expose to our immune system, so that it's trained to stop that virus attack. So that's one component. And the second component is the carrier or the vehicle that then takes that and essentially transports the payload to the bodies for that payload to be presented to the immune system of a human being. So over the last year, we have been looking at various epitopes that make up those payloads, the payload to prevent ovarian cancer. And what we have been doing is that we have been testing how immunogenic these components are. In other words, if you took a blood sample from a healthy individual, took the immune cells from that blood sample and exposed it to any one component of the payload, do these T cells recognize and react and proliferate and object to the presence of that peptide? And we have been screening over 120 of those different epitopes, and we have now almost completed the shortlisting of the best performing ones that are then going to go into the final OvarianVax vaccine. So in parallel with this work, we've been facing a big challenge. I was talking to Faye and Peter about just before we started, where it's one thing screening the components, but then putting them together in one entity, and then hoping that those components will be produced in a cell is really a different matter, and it's a big challenge. So our initial attempts didn't work, failed. However, we screened about more than 50 different iterations to try to come up with a way to produce those epitopes in a cell. And actually, just as of yesterday, we managed to see the green light, so to speak, that it is actually working, that we're managing to get these epitopes together in one entity to form one vaccine. So that has been really, really reassuring. In parallel with this work is then we also started working on the carrier or the vehicle that takes those components and, as I mentioned, delivers them into a human body. So that work is still sort of in its start. But the one thing that I'm also very excited about and want to report to you is that when we take these trained immune cells that have been trained to see these components outside the body, and then put those immune cells next to ovarian cancer cells we obtain from women with ovarian cancer in a tissue culture, what we see is this really impressive response, and that these immune cells seem to be stimulated and proliferate, and they recognize the tumor, while the ones that have not been trained, not been immunized, so to speak, they don't recognize the tumor. So that's very reassuring indeed. And yes, so that's really where we are at with this work. So thank you.
Great, thank you so much, Ahmed. And for those watching, we have a really comprehensive FAQ about OvarianVax on our website, and Ahmed's answered lots of questions on there. So check that out if you do have any questions, but I'm sure some of them will be answered in the discussion. And it's so exciting to see the progress the team is already making, especially with how many epitopes you've worked through and that you're observing these strong immune reactions already. And we're starting to get a sense of how something like this could eventually become possible and move towards clinical trial. But one of the reasons we wanted today's session to go beyond just the science is because developing a preventative vaccine is only part of the picture. History shows us that even promising prevention approaches still face really important questions around timing, uptake, trust, and how people weigh risk and benefit in real life. And also, I just think generally, globally, researchers are facing additional challenges today, especially post-Brexit, post-COVID, and with everything that's happening in the world. I think bringing these discoveries into reality is only getting more challenging. So it's really exciting to see the progress the team's making. And something I think that's especially interesting and difficult about prevention research is that you're often trying to prove that something didn't happen, that you successfully stopped cancer developing in the first place, which leads me to Peter's talk. So to help us explore that wider perspective, he's going to take us through the story of HPV vaccination, and he'll also talk about some of the challenges along the way, including some of the things I just mentioned. So over to you, Peter.
Thank you very much for inviting me. It's a real pleasure to be here and to talk about this. I've worked in cancer prevention for many years, and it's actually really difficult. So at one point I was professor of cancer prevention, but now, because most of what I do is early detection research, so it's really exciting to see what Professor Ahmed is doing in this field, and I think there's a real potential for breakthrough. So thank you. So when I started my career, the whole emphasis on cervical cancer prevention was on screening. And so although Harold Zopphausen, who went on to win the Nobel Prize, had found a strong link between human papillomavirus, or HPV, and cervical cancer in 1983, I think he discovered HPV 16, and then 1984, HPV 18. In the early 1990s, very few gynecologists believed that HPV had anything much to do with cervical cancer, and they've kind of gone down false routes before. But gradually the epidemiological evidence increased, so that in 1995, the International Agency for Research on Cancer declared HPV to be carcinogenic in humans. And then this paper in 1999, which looked at just under 1,000 cervical cancers, they were all squamous cell carcinoma of the cervix, and found HPV present in all but 3 of them. And so they said that this is a necessary cause, that without an HPV infection, you weren't going to get the main types of cervical cancer. So people began to think differently, and then we were also working on screening tests looking for HPV. But in virology laboratories in Australia and the USA, people were trying to make a vaccine. Once we'd established that this is what caused cervical cancer, the obvious thing was, could we have a vaccine against the virus, which would then provide protection? And the idea was to synthesize the proteins that make up the outer shell of the virus. And they successfully synthesized the L1 protein, which is what most of the outer shell, if you think of as a virus as a ball, and so most of the outside of that ball is made up of L1 proteins. There's a little bit of L2, and then inside you've got all sorts of other things. But what the immune system recognizes is that ball. And it turned out that if you make L1 protein, if you synthesize it and you put it together as little pentameres, so 5 strands of it, it then self-assembles into a ball. And so the immunogenicity of the ball seems to be much stronger than just presenting the immune system with the little bits of protein, but actually having the ball, which really resembles the virus, exactly the same size, leads to a much stronger immune reaction. So the first commercial vaccines went into clinical trials in the early 2000s, and the first vaccine gained FDA approval in 2006, and there was a second one that had approval in 2007. And the results of the clinical trials were really quite remarkable. So when you restricted the analysis to the girls who had not been infected previously, the vaccines provided close to 100% protection against infection with HPV type 16 and 18, the 2 types that cause cancer that were included in the vaccines. And we now believe these really are neutralizing vaccines. They prevent infection with anyone who doesn't have it. And the reason why a few people tested positive to HPV after vaccination wasn't because they had got an infection, but simply the virus was present. It was sitting there, but it hadn't infected anything. So it's like you could detect the virus on the surface of a desk, so you could detect it on the cervix, but that didn't necessarily mean there was an infection. And we saw that that most of the people who tested positive after being vaccinated, if you tested them again a few weeks later, they were then testing negative. Whereas if you looked at people who had a proper infection, it persists. So the less good news was that the vaccines only prevent infection, and they don't help individuals to clear an infection. And they certainly can't be used to treat cancer or even pre-cancer that's been caused by the virus. So that meant that the vaccines need to be given before people have been infected, and they work best when delivered in adolescence. So we were going to give the vaccines to 12-year-old girls primarily. And then cervical cancer is a relatively young cancer. Some people get cervical cancer in their early 20s. Certainly it's quite common in late 20s, early 30s. So quite different than many other cancers, which are mostly in people over 50, over 60 years old. But still, you needed to wait pretty much 10 years minimum from vaccinating to start seeing an effect. And so the first evidence at a population level, testing samples from girls who, young women who were being tested for chlamydia screening. And you see on the top left here, that there's this dramatic fall in HPV type 16 and 18 associated with people who would have been exposed to the vaccine. So this, we didn't know whether they'd been vaccinated or not. This is just looking by when they were born, essentially. Also quite a big drop in some other HPV types where we think there's cross protection, and really very little change in the other HPV types. So it looks like the virus HPV 16 and 18 that causes most cervical cancer was almost being wiped out in the population of young women. And then when we went and looked later on, you could see, so we had this school-based program that came in 2008, and it was being offered to 12, 13-year-olds routinely. But in 2008 and 2009, there was also an attempt to vaccinate all girls in school, so up to the age of 18. And that meant that we could start looking, depending on when you were born, from September to August with the school year, we could start seeing. And you can see in these graphs that the red cohorts of the people who were offered vaccination under the age of 15 have essentially no risk of either cervical cancer or CIN3, which is the precancerous condition that's found on screening. And this is what would have happened in the past. These blue curves, the green are the people who were vaccinated in the sixth form, 17, 18-year-olds, and so they're going down. And so we were seeing an 87% reduction in cervical cancer at young ages in those people. So it really did work. It was thanks to the fact that there was really high uptake of the vaccine. So over 80% of the population who were being offered the vaccine had at least one dose. Sorry, over 80% had all 3 doses, and close to 90% had at least 1 dose. And that's important. If you remember what I said about the protein self-assembling into a ball, to a virus-like particle. So it had been that the virologists all thought that you would definitely need more than 1 dose of the vaccine to get this vaccine to work. And initially it was offered as 3 doses and then to 2. But it seems that a single dose provides very good protection, because these balls have a greater immunogenicity than just giving the protein. And there's now been a trial that actually randomized between 1 dose and 2 doses. But a lot of that evidence came by chance from a study in India where they randomized between 2 and 3 doses. But the study was stopped because of ethical concerns about another study. And it was stopped when a large number of girls had just had 1 dose. And so we were able to follow those people up and show that actually the protection that they had was just as good as the ones who'd had 2 and 3 doses. So it means that we can now be thinking about how to actually eliminate cervical cancer globally. We've seen in England, and people have seen in various other countries, Denmark, Sweden, that through vaccination there's this dramatic fall in cervical cancer in young women. It looks like the protection will last for many, many years. So we haven't yet seen any waning protection, but even if that was to happen, you'd simply need a booster. And so the reason why people are so excited about this is because in the past with cervical screening, you needed to screen women every 5 years from the age of 25 to 65 in order to be able to really prevent cervical cancer as fully as we could. And here, the potential is that a single vaccine in a 12, 13-year-old, 14-year-old will give lifetime protection against this cancer. A cancer that used to be one of the most common female cancers in the world. But there are challenges. So one of the challenges that we are giving the vaccine many years before cancers will develop. And so in terms of the cancers caused by HPV, other than cervical cancer, we still don't have any good data to show that the vaccination is preventing those cancers. So there are head and neck cancers, anal cancer. I think it's interesting to look at what happened when vaccination was introduced. So it was done very well in the UK. Part of that was the decision to make it a school-based program. So if you go into schools, virtually all 12-year-old girls are in school, and so you could get very high coverage. There was a girl, Natalie Morton, who died on the 28th of September 2009, very shortly after being vaccinated against HPV. And David Salisbury, who was head of immunization at the time, said, right, immediately we're suspending the HPV vaccination program, and they recalled about 200,000 doses of the vaccine. 2 days later, a post-mortem showed that Natalie had a very large and aggressive tumor in her chest, and that tumor could not have developed because of the vaccine, and that's what killed her. It had nothing to do with the vaccination. And about a week later, the vaccine program was resumed. And I think because the NHS was seen to take this very seriously, they stopped things immediately, they investigated thoroughly. It meant that the vaccine program resumed, and they kept the public trust. In Japan, I don't know why it went so wrong, but in 2013, because there were all sorts of stories about side effects from the vaccine, including chronic pain and muscle problems, the coverage fell from 70% to under 1% over a short period of time. And essentially, Japan suspended vaccination. It was only resurrected 9 years later. So 9 years of women in Japan have not benefited from HPV vaccination because of concerns about side effects, none of which have been shown to be justified or them to be true side effects. So the only established severe side effect of vaccination is severe allergic reaction to the components of the vaccine, very rare, and people are being warned about it, so happening even less often because people often know that they're allergic to these things, and so it's not used. But despite all that, the graph on the right is showing vaccine uptake across London. And you can see that and this is by the year in which women were born. So in the past, we were getting over 80% coverage in most local authorities in London and averaging close to 90%. And then COVID happened and virtually no one was vaccinated in some places. But since then, the average coverage in London has dropped from close to 90% to about 60%, and there are a substantial proportion of local authorities where fewer than 50% of girls are being vaccinated. And that is really worrying, because this 87% reduction in cervical cancer is only happening because we have very high vaccine uptake. And it's true that there will be some herd effect, herd immunity, that because HPV 16 and 18 have become very rare, we have some time to catch up and get the vaccine rates back up before HPV starts spreading again. But if this continues, cervical cancer rates will go up again. So the vaccine is really good at preventing cervical cancer, but it doesn't work in treating either HPV infection or precancerous lesions, and certainly not invasive cancer lesions. So we've got a paper that's under review at the moment. We did a trial to see maybe it would work in women who've just had high-grade CIN3, high-grade CIN excised. So they've gone through screening, they found a lesion, it's being cut out, and if we vaccinated them at that point, would that reduce the risk of the recurrence? So there's a recurrence risk, and in particular, the trial was looking at could it prevent persistent infection with HPV, and it didn't work. So it really does seem to be only a preventive vaccine. People have been trying to make therapeutic vaccines. There was a paper, the Gates Foundation put together a group to say, how should you design trials to look at that? But we're still at the situation where the only HPV vaccines that are out there are ones that prevent infections, which means they have to be used very early on. So, vaccines for cancer is something that I sort of never really dreamed of. About 10 to 15% of cancers worldwide are caused by infections. It's a quarter of cancers in some low-income countries, but ovarian cancer is not one of them. But as Professor Ahmed said, the science has changed, and we can now think about, just as the immune system recognizes viruses as being foreign and attacks them, and just as that can happen with food, you can get food allergies and you get a reaction to it. So could we get a reaction to cancer? And why doesn't it happen generally? So the understanding of why we don't get rid of cancers through our immune system. But these two advances, one is the sort of what I've called plug and play. Professor Ahmed talked about having these platforms for vaccine development, which makes it much quicker and much easier to develop new vaccines. And the second is identifying exactly which antigens are present on the surface of most cancers, and very early ovarian cancers as well. So we can think this should work as a vaccine. It's harder in a way than for a virus. So the virus is the whole cell is the virus. The whole thing that you're looking for, it's not a cell, as opposed to you've got a human cell which has been transformed and is cancerous, if you like, and then on the surface you'll see things which you wouldn't see on normal cells. But nevertheless, that's the hope. If you were going to do things, I think about two different trial designs. One would be really to prevent ovarian cancer. You'd probably do it in BRCA1 carriers, maybe BRCA2 mutation carriers. They have a slightly lower risk before menopause, because those women, the easy way to prevent ovarian cancer in those women once they've reached menopause is to remove the ovary and the fallopian tubes, and then if the organ is removed, then extremely unlikely to develop ovarian cancer. So doing it before then, if you thought that these vaccines might reduce cancer incidence by 65%, then you'd need to randomize about 850 people. If you thought it was being really good and might reduce cancer by 90%, then 400 people would be enough. If you included STIX, which is a sort of precancerous lesion, in the outcomes as well, then possibly you could use about half as many people in the trials. And the other, with the caveat that we didn't get it to work in cervix, is that you say, once we've removed all the cancer that we think we've got, because unfortunately 70% of women have recurrence within 5 years, could we reduce that recurrence? And I think if you had something that would work there, you could do a much smaller trial because recurrence is so much more common, and maybe 150 people. So thank you very much.
Thank you so much, Peter. It was fascinating, and it was so interesting to see in your example the stark contrast between the success of the vaccine program in the UK and Japan. I didn't really know about what happened in Japan, and it shows how public trust affects uptake. So even if the science is brilliant, it doesn't matter at that point if the population doesn't want to take it. And also, thank you for including the possible trial designs. I know Ahmed is thinking about this already, and seeing it like that kind of makes it feel more real, and that it's getting closer. I think one of the really striking things from your talk as well is the prevention breakthroughs rarely happen because of one single discovery. They happen because the science, public trust, healthcare systems, and the long-term implementation all move together over one time. So thank you so much. So we're going to move over to a bit of a discussion now. So I think what's really interesting listening to you both is that you're approaching the same challenge from very different angles. So Ahmed's looking at how we train the immune system to recognise ovarian cancer early enough, and Peter is looking at what it actually takes for prevention strategies to work in real populations over time. So we're going to talk a bit about that. But to start off, I was going to ask you, Ahmed, what's the most exciting thing you've seen so far in the project? But I think you said that. I think the great immune reactions you've been having, looking at your epitopes, is probably one of them. So I'm going to ask, what are you still most unsure about?
Yes, so you're very right. We didn't really expect at the start to see that much immunogenicity to these epitopes, but yeah, what I personally find remarkable is that many of the epitopes that appear to be occur early in the cancer process, as Professor Sasieni is saying, is that we want to be focusing on epitopes that happen really early in the cancer process. Many of those are giving strong immunogenic reactions. I still want to see also that then, extended to reactivity to cancer cells in culture. I want to be able to see not just reactivity, but also actual these immune cells killing cancer cells. And what we have in the lab and what we are working on is that we have a model of essentially taking a normal cell, a non-cancerous fallopian tube cell, and then induce certain genetic modifications to transform these cells into cancer. And the one thing that I'm really hoping to see is whether if we train the immune cells from the same individual from whom we've taken the non-cancer cells, and then co-culture those trained immunized cells with these fallopian tube cells that are undergoing cancer transformation, would we be able to block that? It's not an easy experiment at all, but we are doing it because that would be, to me, a very, very reassuring indeed. And it would mean that if that is the case, it would mean that the earlier we start the process of immunization, the better, to actually, try to mimic what has been achieved with HPV vaccination by immunizing really, really early before cancer transformation.
Thank you, Ahmed. And I'm sorry to put you on the spot, but I think the question a lot of people will be thinking right now is how realistically, how far away are we from seeing something like this tested in people, say, clinical trial?
I think actually the lab work is going really well. We have certain steps that I've outlined in my talk. I think that those we should be able to complete within about 1.5 to 2 years. And then it's really then the point is as well in that you know, as always, obtaining the funding to be able to run the clinical trial is going to be a bottleneck. But I think something of this importance, if we are able to show good, promising results in the lab, then I'm really hoping that we will be able to obtain the funding to go to clinical trials within perhaps the next 2 years.
That's really exciting because prevention is such a long game, but it feels tangible right now. And then Peter, one of the things that really struck me in your talk is how important timing is in prevention. Could you explain a bit more why vaccines like HPV work best before the disease is established and why therapeutic vaccines become much harder?
I don't know if Professor Ahmed might be better at answering that. To me, it seems that our immune system has really developed for against infections, probably, where there is very small amounts. So if what is the total burden of virus or the total burden of bacteria in the body that the immune system has to get rid of? You're not expecting a large tumor to suddenly appear and have an immune system that gets rid of that. And so to be able to fight, to expect the immune system to get something that is already macroscopic, it's a centimeter in diameter, is really probably expecting too much. The other thing is that with HPV, it's sort of all we had to do for preventing infection is to say, what did the outside of the virus look like? Once you've got disease, it's not the proteins on the outside of the virus that matter. It's the proteins that the virus makes that's doing damage. So the outside is just making its own home again, if you like. And then it's all the things inside which start interfering with the normal cellular biology, which means that the cells can start reproducing uncontrolled because these proteins are knocking out the sort of normal safety pathways, if you like. So to be able to get something that's targeting that, which means that some of it is not unique to the virus. And so are you going to have terrible side effects of the vaccine because it's attacking normal cells as well, which is sort of what chemotherapy is doing. We can't get it only to attack the cancer. So it seems a much harder problem once you've got a large amount of cancer there than when you've only got 1 or 2 circulating cells of cancer or not circulating cells. The other, I think, is that we have to worry about where the cells are. So it's much easier, I think, for the immune system to tackle circulating cells than it is to get deep into a tissue. And so will you get the immune response deep within the tissue if that's where the cancer is starting? So I think there's a lot of reasons why it's probably easier biologically to try and do things as early as possible, but it's harder to study it clinically. Because if you've got to wait for healthy people to develop cancer to see that you stop them developing cancer, that's much harder than if you've sort of got people who've got cancer and you say, well, all this work to treat it, and you can start seeing things very rapidly. So, so some people have gone down the sort of therapeutic vaccine because they thought they'd see things much harder, but it turns out to be a much harder problem. Whereas the catch-22 is, even if you do the studies in BRCA carriers, you're still going to have to do pretty large studies. And if you wanted to use in the general population, if you wanted to offer this to all women, the study you'd have to do to prove that it works in that population will be very large. And so I think it will probably depend on what we think we can learn from these more high-risk populations. And with ovarian cancer, you've got the advantage that a lot of the cancers are actually the same. Whereas in breast cancer, the sort of cancers that BRCA1 and BRCA2 carriers develop are molecularly quite different than most of the cancers in the general population.
Thank you. Ahmed, you mentioned the possibility of looking at stick lesions. Could you explain a little bit about what that means and why finding ways to measure them early impacts such an important challenge in prevention research?
Yes, so stick lesions are sort of a short of serous tubal intraepithelial carcinoma, which means they are the pre-cancer collection of cells that are present in the fallopian tube. We believe that these is where the tumor starts, and in women with BRCA1 or BRCA2 mutation carriers, when they undergo risk-reducing surgery by removing the tubes in the ovaries, we can see in about 5% of them those precancerous lesion, even though they present without any manifestation of cancer, but we can still see these precancer lesions. And the question here everyone asks himself is that whether a clinical trial to look at the randomize women to receiving vaccine or no vaccine, and then looking at the frequency of stick lesions in the vaccinated group versus the non-vaccinated group to see if the vaccine is effective in clearing those stick lesions. And actually, if I may, Faye, I want to ask Peter a question about this, because I completely agree with you, Peter, that really it is proven prevention is not going to be an easy task. And I guess in cervix cancer, in HPV vaccination, we needed to wait for many years to look at those mature trials of showing that girls who have been vaccinated did not end up develop cancer, a very hard and clear endpoint. But my question to you is that whether prior to that stage, were there any sort of clinical stage evidence that then encouraged clinical researchers to go on and undertake this massively long clinical trials, or was it all based on preclinical work?
No, so sorry, I didn't explain it well, but the trials were to show a reduction in the precancer CIN3, so like the stick, that
yes.
also included the HPV virus within it. And so the first trial showed that it could prevent infection, and then the trials that led to having national vaccination programs showed that you could prevent the precancer. Now, the difference between CIN3 and stick is that in the screening program, we already had a screening program that was looking for CIN3 and then treating it. And so it became a much easier thing to do that and show that you actually had a reduction. The evidence that there's been this big reduction in incidence and will shortly be publishing to show that far fewer women are dying of cervical cancer isn't from the randomized controlled trials. That's from the implementation, and it really had to be done at this population level. So there was this big breakthrough, if you like, convincing the policymakers and the regulators that it was enough to show that vaccination would prevent the precursor lesions, and you didn't have to wait to show that it would prevent cancer. And had you done that, I'm not sure that, I don't think we would have a vaccine still. And so, over the years, then, the huge numbers of women globally who've been have benefited from the vaccine. We just wouldn't have done it. So I think that has to happen. I think there's, I don't know whether you think your vaccine would treat stick or whether it would only prevent stick. But I wonder whether you could sort of do a xenograph or something, so you could take stick from a patient and put it into some model system and then see whether your vaccine would actually get rid of it, or whether you think it needs to be given earlier than that, before the stick develops, in order to be able to get rid of it. So I'd be interested to know that.
Yes, so our sort of thinking at the moment is that a stick lesion at the genetic level, at the genomic level, is very, very similar indeed to the full-blown metastatic ovarian cancer. Now, it is very conceivable that between stick lesion and cancer, there are other changes that occur, such as, for example, fooling the immune system, immune evasion, and therefore the ability to spread. So what we are doing, we can't take sticks because it's very hard to identify them. Even if you sort of handed an entire fallopian tube, it's very hard to identify live stick cells. But what we are doing alternatively is that we're sort of taking those cancer cells and then building organoid models, which is like a tumor in a dish type, and then looking at the effect of these immunized cells on these cancer cells in the dish. Now, in that way, you sort of trying to overcome immune evasion that is related to tissue barriers and so on, but you can't overcome immune evasion that is inherent to the tumor itself, that the tumor might have developed. So that's the problem with that system that we have. That's the shortcoming. But if you, on the other hand, take that system that I had in mind, where you say, okay, start by a normal cell and try to transform it to become cancer. Can you prevent that process? And if it's established, can you kill those cells? I think that if we get that to work, then that would be very, very encouraging indeed, because you overcome the issue or you have more control of the issue of tumor evasion.
Thank you, Ahmed. And we're running out of time, unfortunately. I'm really sorry to stop the discussion, but just to end, I'm going to ask you both, if we were sitting here again in 10 years' time, say, what would you hope has changed because of work like this?
Shall I start?
Yeah.
Yes. How do in 10 years' time? Okay, I will put my neck on line and say I would love to see the results of the randomized clinical trials. That then we can still, if I'm still there in 10 years, I'm still sort of trying to persuade the regulators to actually to say, well, let's get started with a nationwide and a worldwide vaccination program. Yeah.
Great.
It's very similar, but I think I would hope that in 10 years' time, the vaccine might start being used in BRCA1 and BRCA2 mutation carriers in their 20s or something, and up to when they would like to have surgery, and to start collecting more and more evidence, and maybe people would delay surgery because the vaccine seems to be working, and from that try and make the jump. So I think it's unlikely that in 10 years' time we'd be ready to say this should be used for the general population. But the more we get on the safety of the vaccine and the efficacy in BRCA1 and mutation carriers in particular, I think the greater the demand will be. And it's I've also done work in breast cancer prevention. People like prevention that actually almost eliminates the risk. So a lot of women want to have risk-reducing surgery. Very few women want to take drugs for 10 years to reduce their risk by 50%. And so the beauty of the vaccine is that it's probably one-off. You might need a booster in 10 years' time or something, but you have a course of vaccine and then you can forget about it, and it's providing protection. So I think it really is quite an exciting area, and I just hope that all the next steps they all go well. But if it's not in 10 years, maybe it'll be in 15 years.
Thank you so much to you both. And I think the discussion today has really shown that prevention is so much more about the scientific breakthrough alone, which I know Ahmed, he's not just thinking about the science, he's thinking about the whole pipeline of work. And that's as well why we believe that patient and public involvement has to sit at the centre of this project from the very beginning, which Ahmed's doing as well. And also the reason conversations like this are possible is because supporters continue to back his bold early stage research, which is so important even when the path ahead is difficult. But if anyone's going to do it, it will be Ahmed. So thank you everyone for joining us today and for being so engaged with the OvarianVax programme. I know everyone has been so engaged with it, and Ahmed receives lots of public interest and lots of fantastic questions and thoughts, and he's so appreciative of them all. We'll continue to share updates as the project progresses, and we really hope this is the start of a much bigger public conversation around ovarian cancer prevention. This session has been recorded and will be shared on our YouTube channel soon, which I'll send out to all the attendees. So please do share it to anyone else who you think would be interested in hearing about this work. So thank you so much again, Peter. Thank you, Ahmed. And thank you everyone for joining. And we hope to see you again in the future at another one of these webinars. Thank you, everyone.